What is cellular respiration? Let's break it down. When we think of the word cellular, we think of something corresponding to the cells. But when we think of respiration, we think of something like breathing, involving oxygen and carbon dioxide. Who does cellular respiration? Well, since it corresponds to the cell, that means that all living things do cellular respiration. Let's take a look at the balanced chemical equation. 36 ADP plus 36P plus C6H12O6 plus 6O2 facilitated by enzymes yields 36 ATP plus 6 CO2 plus 6 H2O. Now that's all great, but what does that really mean? Let's take a look. The balanced chemical equation tells us that we want to put together ADP molecules with P, or phosphate, to create the ATP. However, we also have glucose, C6H12O6, and 6O2, oxygen, which we need to break down into the waste products of carbon dioxide and water. Now we're really getting into the heart of cellular respiration. We want to find out what the goal of it is. The goal of cellular respiration is to create ATP. Now you may be wondering what ATP is. ATP, otherwise known as adenosine triphosphate, is usable energy. So the cells can use this energy to carry out life functions and processes. Let's revisit our balanced chemical equation. As you can see, we have glucose as a reactant of this chemical reaction. And we need to break that down. So let's start cellular respiration. There are three parts to it: glycolysis, the Krebs cycle, and the electron transport chain. Let's start with glycolysis. The location of this process is in the cytoplasm of the cell. Remember how we talked about glucose? Glucose is known as C6H12O6. It is a monosaccharide made up of 6 carbons, 12 hydrogens, and 6 oxygens. But for now, we're going to focus on the 6 carbons, as illustrated. One of the 6 bonds is broken in order to create 2 separate molecules, which we will call pyruvates. Glycolysis is an anaerobic process, which means that it requires no oxygen in order for the process to occur. Let's review. First, we invest 2 ATP in order to break glucose down into the 2 pyruvates. In doing so, we gain 4 ATP. If we total that, negative 2 ATP plus 4 ATP, we get 2 ATP, which is our net gain during glycolysis. Alright, now let's start the Krebs cycle. But wait! The pyruvate only moves on if oxygen is present. What happens if no oxygen is present? In that case, the pyruvate does not go through the Krebs cycle, rather it undergoes a process called fermentation. Let's assume that oxygen is present. Before the 3-carbon molecule can enter the Krebs cycle, each pyruvate loses a carbon. These new 2-carbon molecules are called acetyl-CoA. Now we are finally going to enter the Krebs cycle, otherwise known as the citric acid cycle. It occurs in the matrix of the mitochondria. It is an aerobic process, meaning that it does require oxygen. Carbon molecules are sequentially broken off. In this breaking of the bonds, energy is being released and harvested. Let's review the Krebs cycle. When the carbons are being broken off, energy in the form of electrons is released. Harvesting this energy creates a gain of 2 ATP. Let's look at the total ATP we've generated thus far. 2 ATP from glycolysis and 2 ATP from the Krebs cycle, giving us a total of 4 ATP. Remember glucose, C6H12 N6? Well, now we're going to look at the hydrogen. Hydrogen is made up of 1 proton and 1 electron. When we break an electron from a hydrogen, we are left with only 1 proton, which we can call an H+ ion. NADH, an electron carrier, harvests the energy from the electrons broken off during the Krebs cycle. It uses this energy to transport protons via a pump, creating a concentration gradient. Let's look at the electron transport chain. It occurs in the inner mitochondrial membrane. It is an anaerobic process, meaning that it does require oxygen. There is a clear concentration gradient. ATP synthase is an enzyme or biological catalyst that puts together ADPs and Ps to make ATP. H+ ions diffuse through ATP synthase to create this ATP. Energy from electrons powered the transfer of H+ ions to the membrane. A concentration gradient was created when the H+ ions went with the gradient and diffused through ATP synthase. ATP synthase now had enough energy to combine ADP molecules and phosphate, creating ATP molecules. Oxygen is the last acceptor of electrons, and this is what gives the energy to create ATP. 32 ATPs are created during the electron transport chain. Let's look at our total ATP count. We have 2 from the glycolysis, 2 from the Krebs cycle, and 32 from the electron transport chain, giving us a total of 36 ATP, as seen in the equation here.